If you've read Michael Crichton's Jurassic Park, you know you want to be able to turn it off. You want a safety lever in case something goes wrong, and that was me. I was working on a safety lever. Take E. coli as a really good example. Not all E. coli will make you sick, but there's some that are enterohemorrhagic, right, which is a big long word for kills your insides. And we care about that E. coli far more than we care about the ones that are benign that don't hurt us. And so to tell the difference between those E. coli would be important.
In this episode, I interviewed Dr. Patrick Rydzak, a current postdoctoral researcher through the Visiting Scientist Program with the F.B.I. Dr. Rydzak has focused mainly on diagnostics and vector entomology, with his PhD specifically focused on using a combination of PCR and a technique referred to as high-resolution melting to differentiate between several closely related plant viruses. Hope you enjoy. Well, today I am joined with Dr. Patrick Rydzak. It is an absolute honor and pleasure to be able to talk to you because we've worked together before, which is kind of awesome. Right? You're kind of giving this funny look.
When was
I am not associated with you.
Don't tell them that.
And so you did your master's in biology at UT Tyler, right, under Dr. Blake Beckstein.
Correct.
And
yes, sir.
What did you study there?
Well, the Dr. Blake Beckstein's biology lab really could be better classified as a vector entomology lab. And for anyone who's not familiar with that, it basically just means all those insects that transmit the pathogens that cause disease in people, plants, and animals.
Yeah, and so you were working in his lab doing a master's, and then what was your research there?
My research is actually working with fire ants, which are not a vector of any pathogen that I'm aware of, at least certainly not to humans or agriculture. However, we were concerned with management, and there is a pathogen, a virus specifically, called the Solenopsis invicta virus 1, where we were determining if we could maybe use that as a management tool to control fire ant populations here in the United States, which, as you probably know, are an invasive species and do a lot of damage to agriculture and our infrastructure, especially like electronics, things like that. And actually, my research in particular was using something called RNA interference to regulate how much virus was in a colony at a time. So the way Dr. Beckstein liked to put it, which I've always liked, is if you've read Michael Crichton's Jurassic Park, you know you want to be able to turn it off. You want a safety lever in case something goes wrong, and that was me. I was working on a safety lever so we could
for that virus, so you could
for
turn
that
off
virus. Exactly. So we could, so we could better control it. It's always about targeted management of a pathogen or a pest insect.
Did you all ever think of doing, if you had Solenopsis invicta virus 19 and enforced a stay-at-home order on the fire ant colonies?
We tried that. They didn't listen.
They were very Texan.
Yeah, exactly. Within about a day, you get a bunch of signposts that come marching up out of the anthill, and it's just a mess. So it didn't work out too well.
Stingers ablazing. Yeah.
Oh yeah, absolutely. They were not happy.
So what inspired you, or what made you even want to go into that field in the first place?
That is an excellent question, and I would say I more fell into that field by accident. Never once in my life did I ever consider entomology. Nothing against it. It just never came to mind. And it was I was one of those students that fell into, I think, what's probably a common trap for young people, which is you're growing up and you think, okay, well, my parents expect a lot of me, so I should do something like, the big three: doctor, engineer, lawyer, right? And or some combination thereof. And maybe it's not, maybe you don't have a big passion for it or not. Obviously, those are all very valuable, needed professions, but not everyone necessarily has to go into those, right? There's a lot out there. I didn't know that as a younger person.
Yeah.
So as I was kind of figuring out my life direction after my undergraduate degree, I think it was my dad who kind of suggested, why don't you look into these master's programs? And I emailed a couple professors, and Blake, Dr. Beckstein, answered me very quickly, and he got me in, interviewed me. He said, "Great, well, we'd like to have you." And mind you, this was close to the end of the summer, so I was thinking, okay, well, it's a little short notice for me to start this fall, so I'll just keep working, and then I'll start my master's in spring. And he said, no, I want you now. So like a month later, there I was starting my master's degree, having no research experience whatsoever, but he was fantastic and really gave me all the tools I needed to succeed.
Yeah.
As far as why I ended up in fire ants, he had two big projects going on at the time. He had one with fire ants and another with the potato psyllid. And
Mhm.
I didn't know what a psyllid was.
I'm,
No
I'm
idea
sure
what
there are a
this.
lot of listeners right now that don't know what a psyllid is.
Think of it like a very small looking fly. I couldn't give you a much better description than that, but I didn't know what it was at the time. And it was an important study because it was research on a pathogen that this particular insect carries to potato, which was destroying or was damaging the potato chip industry. So it was a very important monetary pest for them.
It's
And
an
again,
actual is a bacteria, right?
that's right.
Yeah.
That's right. Yeah. It causes something called zebra chip, which doesn't actually do anything to make the chip dangerous to you or me, but it makes it have that. You've ever seen those black rings you have in potato chips sometimes? If you have
Hmm.
open a really high quality bag of potato chips, they actually filter those out. But the cheaper brands might not go through that effort because it's not dangerous again, but it's just an aesthetic thing. And it was costing them money, and so they wanted to solve that.
Wow.
And so, having grown up in East Texas, I was very familiar with fire ants. And he said, "Hey, there's this virus that kills them. You want to study that?" And I was like, "Heck yes, we're going to do that." So that's how I fell basically into entomology.
Now, your PhD advisor, Dr. Astri Wayadande, so she's at
Hmm.
Oklahoma State University, and she does a lot of mosquito vector work, talking about how important insects are. That's one of the
Sure.
biggest animal killers on the planet.
Sure. Number one, actually. Yeah. I don't know what's next that, but they blow the next
Wow.
two or three out of the water. It's not even close.
Yeah,
So
yeah,
talking about
yeah,
as far important killers in the world, mosquitoes are top priority.
yeah. So then you went on to do your PhD at Oklahoma State University.
That's correct.
And what did you do your project on there?
So when I went to Oklahoma State, I changed gears a little bit. I went to plant pathology, and basically it's the other half of the coin of the vector entomology. So instead of studying the insect and the pathogen it transmits, now we're looking at the pathogen transmitted to the plant. And my particular field of study was working with viruses of wheat. There's a few different projects I had with it, and I have a feeling you've got questions regarding one of those projects, we'll get to in a little bit. But the gist
Yeah.
of it was I was looking at ways to detect viral pathogens in wheat and then mitigation strategies once we detect it.
So it's really neat. So like you said, we'll get into one of your research papers here in a moment.
Mm-hmm.
And it's very interesting because I've found now in a few of these different researchers that I have this conversation with is, when they are talking about viruses and within a host, it's very relevant to what's going on with our global pandemic right now, and
Sure.
detection of viruses.
Mm-hmm.
But before we jump into that, I want to ask, because, you had this really cool path, even though, you say, you didn't know you wanted to do entomology and you kind of just fell into it. You've kind of, you've done very well. You kind of
Oh,
flew through your
thank you.
master's, you flew through your PhD, and now you're working with it's called the Federal Bureau. I think it's French. Federal
That's right.
Bureau,
I'm sure it's a
of,
French
of investigation.
French
Yeah,
origin
you work.
for that word. That's right.
Yeah, so you work now with the FBI, right?
That's correct.
And
Yep.
that's not everyone just gets a job at the FBI.
Mm-mm. No, so specifically my job title is I am a postdoctoral researcher with the Visiting Scientist Program, and we do different research projects specifically in forensics. And you're absolutely right, not everyone gets the opportunity to do work with an organization like the FBI. As you can imagine, their standards for hiring are very high. There's a lot of extra hoops you go through necessarily to get a position there. But the researchers who work there and the work they do is of the highest standard I've currently encountered. And that's saying something. I'm not demeaning the people I've worked with in the past when I say that at all. I've worked with top caliber researchers
Oh
in
yeah.
these agricultural fields. But once you get to a forensic level, we're talking about things going to court possibly, the standards are maxed out from bottom to top, all the way from the start of projects and how you record your data, all the way to when you're writing your articles, presenting the data.
Yeah, I
So
mean, I remember when you were applying, I was visited by an FBI agent, and she had the seal, Federal Bureau of Investigation
sure.
on her handbag and everything, and very professional. And I was just kind of surprised that there was someone that came all the way to East Texas, to this AgriLife
Sure.
center there and it makes sense because you're working at the highest level in terms of security.
Mm-hmm.
And, but I also know, so you say that you're doing a lot of kind of, you say you're a visiting scientist. So what I know from TV shows means that you're also out in the field catching the bad guys, probably, and throwing your
No.
badge around, taking cars and riding boats and stuff like that. You don't have to tell me. It's fine. It's fine.
That's right. I can't tell you about anything related to that, mostly because I don't know anything about that. I am in a laboratory just like you, just like all these other researchers. We just have different focus of research.
That's awesome. So did you always want to work for the FBI?
It's something I had never considered. Growing up, I always thought it was really cool. I was one of those people who, like I'm sure a lot of people my age, grew up watching X-Files. Some of my favorite novel series include FBI agents, and they're always very intelligent and doing really cool things. But I just always assumed, oh, okay, to do that you have to be a police officer, right? You have to be law enforcement. You have to be someone with a badge and a gun. To go and work for an organization like this. And actually it was Dr. Beckstein who kind of put the bug in my ear one day. I don't remember, I know, right? Entomology and, well, what can I say? And he kind of put the idea in my head. He said, have you ever considered working for the FBI? And what do you mean? I didn't know that's something I could do. Oh, they have scientists there? What do you mean? And so that's how I kind of started on the path where I ended up at Oklahoma State University, because they had connections to the FBI through what used to be known as the National Institute for Microbial Forensics, Food and Agricultural Biosecurity. Take some practice.
You remember that acronym?
Oh yeah.
What's the new one? Do you know what the new one is?
And it's much shorter. That's so went from
I am F.B.I.
Something like that. But basically, microbial forensics and agricultural biosecurity. Smash those two together, and that's what their focus is. Mm-hmm. And that's, of course, very important field in terms of keeping our food safe and secure, which are two important components of keeping populations fed and happy and able to go about their jobs and not worry about their families and things like that.
So how did you find the opportunities to join the FBI? I mean, if someone is interested, how did they do that?
So originally I came to OSU on a National Needs Fellowship, with the idea being they're looking to fund PhD students who then move into biosecurity fields. In the government, although it wasn't required we go into a government job, there are some fellowships and scholarships that are like that, where you're obligated to a certain field. It wasn't like that in this case, but it was kind of my interest anyway at that point, so it wasn't a big deal. And as a requirement, we're supposed to do an internship somewhere, and they gave us some money to do that. And I got notified about something called the FBI's Honors Intern Program. And essentially what this was is an FBI attempt to pre-screen a lot of potential employees for their future ranks by targeting younger people. So we're talking about undergraduates, master's students, obviously me, PhD level at that point. So all different fields, which about law students, biology students, chemistry students, you name it. They were advertising to these students, and you could apply. And then after, if you were accepted, after a background investigation, then you'd be brought into the program, and you'd be doing any number of different things. So it was a really great opportunity, and I actually ended up working in the laboratory. And so that's how I got introduced to the laboratory. And I was like, this place is great. I love what I'm doing here. And then when I had the opportunity to come back, when I finished my PhD, I applied for this visiting scientist program, which again is a contract position through something called ORISE. It's Oak Ridge Institute for Science and Education. And I was very grateful to get the job I have now.
All right. Very cool. And was there a reason why you didn't consider either academia or private industry as opposed to this? Was it just because you were just super passionate about it, or?
And so to tell you the truth, that I haven't dropped any considerations at all. What drew me to the FBI in general, or you could say government in general, was, for me, and I'll frame it this way. there's, in my opinion, two kinds of researchers. There's those that are just, from the day they're born, they're just passionate about one thing. They just have to study that one thing. I have to study the mating behavior of the African blowfly. I have to do that. I won't be complete in life. I'm not a complete person until I do this thing. And those people are awesome. And they are the kind of people that lay the foundation for the work that I'm interested in. Is once we have that baseline information, data, facts, theories, what can we use? How can we build upon those to improve our lives, or to improve the lives of agriculture, or improve agriculture, improve the lives of animal welfare, whatever it may be. So I'd say I fall into that second category.
All right.
And the FBI, as well as, you could say this, as well as industry, they take that baseline knowledge that's generated through those first kind of academics I was talking about, and they apply it towards something that's really tangible, and I can see the results now. I know exactly how what I'm doing really is helping someone down the road.
I'd say I'm just the exact same way. There's that discovery-based science, like you're saying. They're making new discoveries, but it's not always very applicable. And there's us applied, yeah, at least not immediately. And then there's us applied scientists then that might take some of that work and think about how to actually apply it and get to see those real, world results. And that's, for me, what is my passion, is not just the science, but also seeing how it ends up directly impacting people.
For sure. So many of the greatest discoveries we have in science were accidents, happy little accidents, as Bob Ross would say. And my favorite is one in which, I think this was in the 80s, but a team was trying to make a more purple petunia. Okay. Yeah. Perfectly valid and reasonable thing to, sure. Yeah, absolutely. I love purple colors. Fantastic. I sent my mom some purple flowers for Mother's Day. They're beautiful. So I understand the draw very well. And so they knew, okay, we have this molecular piece, this molecular blueprint that codes for purple. So if we put more of that into the plant will make more purple. That makes perfect sense, right?
Yeah.
So once they did this, not only did the plant not make purple, the plant actually turned completely white. And so what had happened was the plant, and this is a technique, or something called RNA interference. And what was basically happening is the plant saw this exogenous RNA is what it was, and they said, oh my gosh, this is a virus. It looks just like a virus. Kill it, and kill everything that looks like it, anything that even resembles like anything close to it, and just get rid of it. And that's what it did. And so it knocked down that purple, that code for purple, and it also knocked down its own code for purple. And since then, we've used this technique, which is basically the birth of a lot of our current genetic engineering, the progenitor, as it was, to some of the bigger name ones you might have heard, like CRISPR-Cas, right, which is like the genetic scalpel that's always in the news. Yeah. So you never know where those little accidents will end up in the 20, 30 years in the future.
All right. So now I thought we would discuss your paper,
yes,
which is entitled Combining Multiplex PCR and High Resolution Melting for the Detection and Discrimination of Arthropod-Transmitted Viruses of Cereals.
I am so sorry, by the way.
Beautiful title.
My PhD advisor told me, Patrick, you're really great at what you do, but man, you're awful at making titles. And she's right. I'm sorry.
It's so catchy. I read that title and I'm like, oh, I gotta read this.
I just know exactly what it is, and I'm just driven. No, I'm not
pulled.
It is accurate to what it is, but that is all it is.
Yeah. I mean, I read about the low resolution melting, so I have to read about the high resolution melting now. I mean, I'm just teasing. But this is published in 2020, Journal of Biological Methods, and this is it's actually a really neat paper because it's a methods paper on a technique to essentially detect potentially multiple viruses at very low dosages within plants. And you have here Dr. Francisco Ochoa Corona as a co-author, who is like a diagnostics specialist. Like that's what he focuses his research on, right? Is like field diagnostics and enhancing diagnostics. Is that right?
Absolutely. He's one of the premier viral plant virus diagnosticians in the world. And I had the great pleasure of learning how to do PCR and primer design, which we'll get into a little more what those words mean in a minute. But he was the one who taught me that, and I was very grateful because he's excellent.
Yeah, yeah, I've had a chance to meet him, and just a super nice guy. And again, some of the stuff they're doing is really cool.
Hmm.
And so you're looking at some viruses of cereal crops such as maize, wheat, and barley.
Hmm.
And in Oklahoma, in your paper, you discuss how wheat is one of the state's most important exports,
Hmm.
with about $471 million in 2016 alone, is what it was valued at.
Hmm.
Whereas these viruses, just one of them, the wheat streak mosaic virus, can cause losses up to about $500 per hectare,
Hmm.
which can amount to quite a bit.
Profit margin on the wheat is incredibly low. And so anything that cuts down that profit margin for the growers almost doesn't make it worth them even harvesting in the first place. So we're talking about it's even more impactful than what you might think.
Yeah, so that's kind of funny that you mentioned, because you have a few numbers in here, right? There's 5 million acres valued at $471 million in 2016 alone,
Hmm.
and then you have this wheat streak mosaic virus alone causing losses up to $464 per hectare. And if you run the
Hmm.
numbers, it's like if all the acreage or all the hectares had this virus, I mean, they're all running at a loss. I mean,
Oh yeah.
it's like, yeah,
Absolutely. Hmm.
yeah,
And
yeah,
what you see actually, it's interesting. I didn't know this until I started going out to the field to see these and meet with these growers. But in years where the value of wheat is low, you actually have higher incidence of viruses the next year. And you're thinking, what on earth does the monetary value of wheat have to do with the amount of virus that's there the next year? Well, what turns out is what happens if the wheat's not valuable enough, then they don't bother harvesting it.
harvesting.
That'd be a cost to them. So it sits there, and it is a reservoir then for that pathogen the next year, which so it just makes it expand there quicker and spread through the other fields faster that way. So it's just
Yeah,
things you never think of. And
right.
so talking about a chain reaction where you have low value because of damage from virus leads to crops not being harvested, which then leads to the next year even lower values because there's more pathogen pressure. It's just
Gosh.
it's yeah, it adds up more than you'd think.
And that's why, I mean, if possible, and sometimes it's just not feasible, but it's encouraged to rotate crops, and you're like rotating between completely different families or groups of plants so that they're not able to cross transmit certain viruses. But
Mhm.
if there's no market, or your soil type or your conditions are not well suited for legumes, or
Sure. Yeah,
whatever,
exactly.
yeah,
Mhm.
then
We're
it
talking about
doesn't
the
really work.
panhandle of Oklahoma. There's what you see is what you get.
Yeah.
So you don't have a whole lot of options. I think canola is another big crop that I see often grown there as well. So they have some options, but really not too many.
Yeah, and sometimes, so there are several different viruses that these plants can get, and sometimes you get these co-infections with multiple
Mhm.
viruses. So your paper is looking at trying to determine kind of what are all the viruses that are occurring there, especially in co-infections as well, and why
Mhm.
is that? Why is that particularly challenging?
Yeah, good question. So co-infections are particularly scary. In a lot of cases, they'll synergize. And what I mean by that is you can have a sick plant with just one virus, and it might still be a little engine that could and still grow out, and we can still harvest from it, maybe just with lower yield, things like that. But in some cases, when you have another pathogen that's also infecting the same plant, then that plant just can't handle it and it just dies. And we actually have, just off the top of my head, I believe it's Triticum mosaic virus and wheat streak mosaic virus. They're both from the same family of viruses. And when they're both found in the same field, then you can have crop losses of 100%. So the
Wow.
entire field gone. And that's opposed to if there's one or the other by themselves, you can still harvest and you might just have a little bit of loss. But when they're both together, bam. Another really good example is in Africa, they're struggling with something called maize lethal necrosis. And what that is, it's a syndrome that's caused by a synergy of maize chlorotic mottle virus and a potiviridae. And a potiviridae being the virus I just mentioned, wheat streak mosaic virus is one of them.
Okay.
So with that combination, it just decimates the maize. And of course, maize is such an important crop for them. It's one of their greatest food sources, the greatest food source, I think there. So if they have big losses in crops, they just don't eat.
That's
So
lack
you
of food
see how
security. That's a food security issue there.
exactly, it's a very big food security issue there. So that's why we really care about determining how many viruses there are in a field at one time and which ones are there, because it can
Yeah.
make the difference between having a harvest and not having a harvest.
And so oftentimes when it comes to, virus detection or detection of any type of microorganisms, PCR or polymerase chain reaction is a common technique that's
Yep.
often used.
Mhm.
And you did a lot of PCR. What is PCR?
I've done a lot of PCR, absolutely. PCR stands for polymerase chain reaction. And actually, I thought this would be a good moment to say PCR was actually invented in 1983 by Dr. Kerry Mullis, who was a chemist. He actually just died last year in August.
Wow.
And
Wow.
it's important to mention him because this person revolutionized the way molecular biology is done, period. Actually, I think the New York Times wrote it best, and I'm going to paraphrase. Basically, they can say there's a time before PCR and a time after PCR. That's just that's just
Yeah,
how it is. And so
absolutely.
what PCR is, it's basically a method for copying DNA. And we're talking about copying a very, very specific, very small piece of DNA. So the example I like to give is imagine how your fingerprints might be a very small part of you, but yet from those very small parts, we can identify you compared to anyone else in the world. Okay, so the same thing with DNA. We're looking at a very, very small piece of DNA or RNA, and we'll get into the difference there in a little bit. And from that small piece, we can identify, okay, this is this pathogen, or this is this bacteria, or this virus, whatever. And so how PCR works is you have an enzyme called Taq polymerase, and Taq polymerase just copies DNA. That's all it does. Doesn't have any idea about what DNA to copy until though you tell it which piece of DNA you want to copy. And to do that, you use primers. If you want to think of primers like bookends, you put these bookends on two ends of the DNA you want to copy. Taq polymerase says, okay, that's what we're going to copy, and it copies that spot over and over and over and over again. And all of a sudden you have go from a couple copies of this piece of DNA to thousands to millions to billions. It just goes up exponential.
And so it's using this. I guess another term for it might be a temperature cycler. Is that a common term for it or not?
Yeah, so we use a thermocycler to have
Thermocycler.
this. Yeah, to have PCR occur, and basically how it works is at different temperatures you have the DNA break apart. You have the primers bind where you tell them to. Then the enzyme Taq polymerase finds those primers, those bookends, copies that piece of DNA. Then we heat it all up again, and everything falls apart, comes back together, and
Yeah, so
repeats
you keep
over and over
heating
again.
off and cooling down, and with that's when that either things break apart, and when it cools down, then things can come back together and create a new copy. And that's how you keep splitting the DNA, and then allowing those primers to bind, and the Taq, basically fills in the spots. And the
Exactly.
primers would require that you have some knowledge about the DNA of what you're trying to replicate. So you need to know those bookends. You need to know, we're talking DNA sequences of, however many 10 base pairs or 5 base pairs on each side. How many is a primer usually?
Primer's usually 20. Sometimes
20.
it can be longer or shorter depending on what you're trying to do, but 20 is generally the minimum.
Yeah, and so so that primer is designed for the beginning and the end of what you're trying to replicate, and you'll get copies of everything in between. Is that right?
That's exactly right. Yep.
Yeah. And
And
so
then sorry, go ahead.
Sorry. So why is that alone? That seems like it'd be adequate enough to replicate the virus and to find out what it is. So why is that? Why is PCR alone insufficient?
So PCR alone, even though we're talking about billions and billions of copies of this small piece of DNA, it's not like you can visually see that. We're still talking about a very, very tiny tube with a little bit of clear liquid in it. It doesn't matter if you put it under a microscope, you're still not going to be able to see DNA being copied in there. So you have to add an extra step to be able to identify if anything's in there. If your primers found their target, if your target was there, then the DNA was copied, then you have all these extra copies floating around, and they're a very specific size. And so from there you use other techniques. One is gel electrophoresis, which basically just filters out all the DNA of different sizes. And if you have a lot of DNA of one size, which is what you get if you have your target and PCR works right, then you'll have a very strong white band there. You'll be able to visually see it. Okay,
Hmm.
so that's gel electrophoresis. Then there's other techniques called real-time PCR is a very, very common one. We can get into more details about that. And there's a couple others, but those are probably the two big ones.
And so in your paper, though, you discuss how, the PCR, and is it perhaps because you need too many primers, or why is it that you're looking at RT-PCR instead of just
Hmm.
using PCR to look at these co-infections? What's the difference there, and why did you need to develop this new technique?
Okay, good question. So, thank you. I did not develop RT-PCR, though I'm
No, you
flattered.
did. No,
Flattered.
congratulations.
I'm
No, no,
flattered. You think I did?
no,
I
no.
should.
I didn't think you did. I'm saying,
I
why did you think to use RT-PCR instead of PCR for this specific purpose?
Sure. I'm just teasing you. Yeah, I'd be in a much bigger apartment if I had invented RT-PCR.
Or not.
RT.
Us scientists don't always get paid off for.
Yeah, maybe I don't care. I have a bigger fish tank. I'll say that much. But so RT-PCR stands for reverse transcriptase PCR. And I alluded to this a little bit, that there's RNA and DNA. Turns out viruses are predominantly RNA. And for the sake of this particular talk, RNA and DNA are very, very similar, except in RNA, instead of a T, there is a U. All right, so DNA is ATCG, RNA is AUCG.
Yeah.
Right, and that causes a lot of very interesting differences between the two, which gets kind of technical. But the important thing is you cannot actually do PCR on RNA. So you
Oh,
actually have to
interesting.
do you have to do something else to it first, and that extra step is called a reverse transcriptase reaction, which is where that RT comes in. So
Mhm.
RT, reverse transcriptase PCR, RT-PCR. And so basically what you're doing is you're taking that RNA and you're turning it into what's called complementary DNA, which is DNA that is complementary to the RNA strand that you started with. And so now we have DNA, that complementary DNA we got from our viral RNA. And now we can do PCR on it. Now we can do gel electrophoresis. Now we can see if our virus is present in that sample.
And talking about viruses, so SARS-CoV-2 that causes our COVID-19
Mhm.
is an RNA virus.
It
And
is.
so in order to detect it, right, they got one technique, there's this one paper that's in preprint, uses RT-PCR. So they're making
Mhm.
that complementary DNA, and then they're running PCR on it, and then they're doing that, the gel electrophoresis, and they can
Mhm.
detect as low as 5 copies. So there's five
Yeah.
copies of that RNA in the sample. Now, mind you, they said that they need 172 primer pairs. I mean, I guess it's to catch all the different variations, or
So
why?
I'm not familiar with which particular study I have not read that article, but my guess is in that case, I think they must be doing some kind of population analysis. That many primer pairs is overkill for just detecting your virus. I did a
Gotcha.
little bit of digging on it, and most of the studies I've seen, they use 3 different primer pairs put into one reaction. It's called a triplex, right? Tri
Okay.
for 3, plex
Hmm.
PCR, and they're targeting just 3 sites on the virus. And they target what are known as conserved sites, or sites that don't change very much. So with RNA viruses in particular, every time they replicate, they're not really good necessarily at replicating exactly right. Their proofreading isn't very good. So a lot of um, changes are made to the genome. I think for every 10,000 base pairs, you get one what's called SNP, or single nucleotide polymorphism, which is where you have one nucleotide
Change.
changed to another nucleotide. And these can be in random places, all through the genome. But what's really important is with these conserved regions, basically when you think of conserved, I want you to think of important. So important for the function of the virus.
Hmm.
So if too many changes occur in these conserved regions, the virus can no longer replicate. And just for some framework, we're talking about viruses which are 10,000 base pairs or 30,000 base pairs in the case of coronaviruses,
Yeah.
about 30,000 base pairs. They're the largest RNA viruses. That's really not many nucleic acids to work with. Any
Yeah.
changes to that many within that many base pairs is pretty significant. Um, so you target these conserved or important regions for your PCRs, so that you're more likely to catch all the viruses, all they're known as quasi-species. You have all these different viral genomes that have SNPs here and there. So to be able to detect all these different quasi-species, you want to target those conserved regions.
Because those are the ones that are still going to be, in theory, I guess, functional. Is that right?
Sure.
Or
Yeah,
yeah,
absolutely. Yeah. Otherwise, they won't make it. They won't be able to replicate after that first time if there's some really critical error in in the replication process.
right. And so, what are some of the limitations of reverse transcriptase PCR, and what method did you look to combine with it to decrease the number of primers used? So, I guess number of primers is an issue, or
Sure. So one issue is if you're one, this PCR thing, it's awesome. You can detect all these different things. So why not just slam these tubes we're using PCR with full of primers, right? We want to detect everything. Well, the problem is once you do that, now you have these two primers, there's these two bookends, right, that have to work in concert with each other to just target the two ends of exactly what we want. But if you have all these different bookends floating around, sometimes some of the bookends kind of look like they go with this bookend. I'm not really sure. Maybe that bookend goes with this one. And through just the thermodynamics, you can get some really funky things happen just through entropy, randomness.
yeah.
And so we want to limit the number of primers we use in a reaction just to reduce that chance of having what's called off-target amplification, or something else called primer dimer, which is where the primers like to match up to each other instead of the target genome you're looking at. And then from there, Taq polymerase just, okay, I'm going to do my
Keeps
job.
replicating that.
Yep, and
Yeah.
I'm going to copy anything that these primers are next to, and it doesn't matter if they're touching primers or where they're supposed to be.
Yeah.
So we want to limit that number as much as we can.
Yeah.
And so when you're talking about these assays that have hundreds of primer pairs, those are very difficult to do, and my hat's off to the people who manage those.
And so you have the situation where you're trying to detect there's like 3 main species of viruses, but then you also have some subspecies. Like how many different things were you trying to detect in total?
Sure. So in my essay, I was looking at 3 predominant viruses that were important to the Great Plains region of the United States, with this big kind of S shape that goes right up through, starting in Texas, all the way up Wyoming. And we're looking at wheat streak mosaic virus, maize mosaic virus, and barley yellow dwarf virus. All 3 important viruses. Now, one of those viruses, the barley yellow dwarf virus, is very interesting. Its family tree is really convoluted and hard to keep track of, and I spent hours on my PhD thesis trying to get it right. Still got it wrong, didn't matter. But to save you the brain strain, there was 3 species of that virus I was interested in. Each had their own designation. So they all started with barley yellow dwarf virus, or BYDV dash something. In this case, it would be PAV, PAS, and MAV. Now, again, you could add more primers to detect those different species, or in my case, what I did is I went to something called high resolution melting. So I developed something called Degenerate primers, which, don't worry, they're not like bad primers, degenerates.
They're useless.
They're just absolutely no morals whatsoever. No, so what Degenerate means in terms of primer design is that instead of a nucleotide that's just an A, T, C, or G, you have another letter you put in there. And it's not an actual nucleotide you put in there, but it's a
Like a
letter
wild card.
that exactly, a wild card, a great way to put it. It's a nucleotide, it's a letter that corresponds to different nucleotides. So for instance, you could have a series that starts with C, A, R, T. Now, R, if you notice, is not one of the 4 nucleotides that make up DNA.
Right.
R stands for, I believe off the top of my head, an A or a G. So you can have CAAT or CAGT, right? And it's going to be one of those 2. And so if you have a primer then that has an R in it, that means you're going to have one primer sequence that has the A, and this identical sequence, but instead of the A there, it has a G there.
Yeah.
Yeah. So we developed these degenerate primers, and the purpose of doing that was that then what we can do is, because all 3 of these species have different slight variations in their genome, right? Those SNPs we talked about earlier.
Yeah.
We could use the same primer pair, right, with those slight modifications in them, and then we could detect all 3.
Or at least you could replicate all 3.
Yes,
right?
exactly.
Yeah.
Exactly. Yeah. Very well put. Yeah, exactly. You could replicate all 3. But the issue there now is we have 3 different sections of DNA that are all the same length. Now, if you're using gel electrophoresis, you're identifying your amplicon, your PCR product, by its length. So if we have 3 different targets that all have the same length, that means you can't tell them apart just
Right.
by looking at the length. So that's where you have to bring in another technique, one called high resolution melting, which is really metal. And what you're doing is you're just applying a lot of heat to your PCR products, those short sequences, until they melt. Now, what's really cool is with these really highly calibrated thermocyclers, you can apply heat at a degree of 1/4 of a degree per second change.
All right.
So
Nice.
very, very small changes in heat, and you slowly increase the heat 1/4 of a change, or maybe even 1/8, right? Very small increases at a time. And because the thermodynamics between those 3 targets is different because of the SNPs within the sequences, right? So
So we're saying,
different.
yeah, go ahead. Sorry.
No, no so the different letters all match up to each other and have different strengths of how tight they match to each other. So A and T match kind of well, but G and C match really well. So if you have a sequence that has a lot of G's and C's, it's going to be a really, really tight fit, relatively speaking, from thermodynamics. So not really like Legos, but just how well the bonds are. So versus if you have one that has a lot of A and T's, then the fit's not so strong. So based on that slight increase in temperature over time, you have these bands slowly break apart, and we can distinguish them now from each other by the rate at which those 2 strands of DNA break apart.
That's awesome. And it's like a fluorescence that occurs, right? When they split apart. Is that what you guys were looking at?
That's right. So when you're working with quantitative PCR, or sometimes known as real-time PCR, you include some extra molecules into your reaction which incorporate into the DNA in some form. In this case, they're called intercalating dyes. So what happens is during the process of PCR, you get more and more double-stranded DNA that gets copied and turned into more double-stranded DNA, et cetera. During that process, we have a molecule that only fluoresces when it's incorporated into double-stranded DNA. So if you have more double-stranded DNA, you get more fluorescence. Now, as PCR goes, you get more and more fluorescence until you get to the peak of your reaction. And then from there, if you start melting the DNA, you have that double-strand DNA, if you start melting it, start breaking apart all those fluorescent molecules that only activate while in double-stranded DNA now dissociate from them and no longer fluoresce. And so you get a decrease in fluorescence from where you started. And it's that decrease in fluorescence, the rate at which it changes, that then you can measure, or you can determine which PCR product you're looking at.
Wow. Yeah, and from what I remember, so like, just going back to how kind of that melting works, that A and T, they're bond together by 2 is hydrogen bonds, right?
That's right. Yep,
Just,
2 hydrogen bonds.
yeah.
Yep.
Whereas C and G are 3 hydrogen bonds. And that's
Correct.
why, they basically have a higher, it's a little bit more, they're bonded together a little bit stronger. So they need a little bit more energy, a little bit more heat in order for that to split apart. So the more GC ratio you have in your DNA sequence, the higher that melting point is. And so you can see, so there's like SNPs, right? Single nucleotide polymorphism. So like a single difference between 1A and 1G, and you can see a difference in melting point.
Yes, absolutely. It's
That's
very
incredible.
difficult when it's just one SNP, but it's possible. But like I said, it's difficult. Usually, you need a few more than just one to really be able to tell them apart. Otherwise, there's with enough error in the system, you can't really be confident. But if you have a few, then you can definitely tell, and we could definitely tell in this case.
That's remarkable. That's
Yeah.
cool.
So to summarize again, basically, we target this barley yellow dwarf virus. We take that chunk of DNA we get, then we melt it, and based on at what temperature it melts, its melting point, we can identify which species of barley yellow dwarf virus it is.
Yeah. So in that way, you're using way less primers, but you're using primers that are a little bit less specific, let's say, I guess. They're replicating more things, and then you can determine what things it's actually replicated by using this high resolution melting.
Yep,
That's really cool.
absolutely. It is very cool. And the reason we care about which species of virus it is, if I were to tell you I have a cat, right? Maybe I'm talking about a fluffy domestic house cat, or maybe I've been watching too much Tiger King and I've got, Simba chained up in my kitchen somewhere. So there's a big difference when we say cat, right? There's a big difference between a tiger and a house cat. So we want to be able to tell a difference in some cases with some of these pathogens. Take E. coli is a really good example. Not all E. coli will make you sick, but there's some that are enterohemorrhagic, right? Which is big long word for kills your insides. And we care about that E. coli far more than we care about the ones that are benign that don't hurt us. And so to tell the difference between those E. coli would be important. In this case, with viruses, with the barley yellow dwarf viruses, which virus you have is dependent on which aphid vector is actually transmitting the virus. So we'd have a better idea of which aphid we have to target. So for instance, if you're a scientist who's interested in, I don't know, beneficials, right, for some biological, just throw one
Let's
out
just
there.
say,
Yeah, as an example, I don't know who would do that, but if you have
this
a
guy.
if so, you have someone like yourself who may have an idea about, well, this particular parasitoid wasp targets these particular species of aphids at a higher success rate than other species, and maybe that's important information. I don't know if that's the case. I defer to you for that.
No, absolutely. Yeah, if they are the main target pest, you want to make sure that the beneficial predator that you're releasing is targeting the main pest. If it's like eating other things instead, then that's problematic. So I guess in that same way, it's really important for you to detect, what is the main actual virus in there, and is that the one of concern? Is that the one that you need to, and how to deal with it from there? And so in your experiments, you have both positive and negative controls for detecting the virus. Why is that important?
Sure. So like I said before, we want to make sure if we're trying to detect tigers, we want to be able to detect tigers as opposed to house cats. So what we would do is we design a test that differentiates between tigers and house cats, right? For instance, we would also want to make sure that our test does not target dogs, right? Because we're not interested in dogs at all. Mm-hmm. So we do something similar with viruses. We have positive controls. So for instance, we know that this plant sample has this virus we're interested in. And we do that by testing it using other techniques, and we confirm it. Usually they're sourced from companies that is their function is that they put together these positive controls and they're quality control tested. So we know exactly what's in there. We know it's positive for what we're looking for, et cetera. So that we make sure that our assay is targeting what it's supposed to. But we also do tests with negative controls, as well as other what might be called near neighbors, or other things that look like our target but aren't our target, right? So that'd be the house cat looking like a tiger. We want to be able to tell the difference between the two. So you include in your assay other closely related viruses to make sure that your assay is not targeting those other viruses we don't really care about. Maybe a more applicable example is when they're testing for corona right now, they really want to make sure they're not getting influenza. Right? We really want to make sure they distinguish between someone who's got corona versus someone who might just have the flu. Very important distinction there, and we care the same way when we're talking about plant pathology.
And at the same time, so they also want to make sure, and oftentimes this is what I find kind of interesting, is you don't usually probably have any positive control or positive check when it comes to testing for coronavirus. You want to make sure that when you're testing someone, that you didn't miss the virus due to the sampling technique or due to degradation before it actually got to the lab. So you have a number of potential false negatives. It comes back as, oh, you don't have corona, but they very well may have, because we don't have positive checks with all these diagnostic tools that are being used.
Yeah, no, that's a very, very good point. And another side of that is we want to make sure that the assay we're developing, the diagnostic tool, can actually detect, we know what level it detects at. So we do what are called sensitivity panels, where we know the amount of concentration of virus in our sample, and we make it lower and lower and lower and lower until we can't detect it anymore. And that stage that's the limit of detection. And so, for instance, if we're applying that to corona, we have someone come in and we test them for coronavirus. Well, it's like, well, we can't detect it. That doesn't necessarily mean they don't have it, but it's at such low levels we can't detect it. And those are two important distinctions to have. And we want to make sure we can detect it and we don't get false negatives, so we don't accidentally send someone to go back to work, who might then infect some of their coworkers or family.
Right. And this kind of blew my mind, because to determine the sensitivity of your diagnostic tool, you did a serial dilution down to 1 femtogram per microliter.
Yeah.
1 femtogram. I had to look that up.
Sounds like I'm making up words, right?
Yeah, because I think the lowest I had been familiar with, at least in day to day, was like nanogram, maybe.
Sure.
And femtogram is 1 × 10⁻⁸.
I think that or 15th. I forget, but it's a lot of zeros before you get
It's
to
a lot of zeros.
a lot of zeros.
Could you detect it down to that small?
Yeah, that's generally the standard when you design a PCR to detect. And we're talking about that particular range. We're talking about gel electrophoresis. So we're talking about a visual representation. When you go
Wow.
to those quantitative PCRs, we're talking about from there you can actually measure transcript copies or the number of copies of your target within the sample. So for instance, if you have a sample that has 10 virions, so 10 viral particles within the sample, we should be able to detect 10 transcript copies of that virus if your
Wow.
assay is really fine-tuned, and many of them are. I think with these COVID-19 PCRs, I think they're detecting, I think you said 5. I read something that was like 3.8 to 5.5, some range like that. So
Gosh.
really fine-tuned sampling.
That's pretty incredible. Well, I thought that was a really cool paper.
Oh, thank you.
Really neat research. I'm really happy for you for all the work you're doing. Thank you so much for your service. For serving
No,
us
thank you.
Americans.
I'm doing the same job that you and all the other researchers are doing.
That is what a superhero would say. That is.
Yeah, but then they would like bounce out through the roof, and some really cool with the cape flying. I'm not going to be doing that.
You're a modest superhero.
I'll be rolling out of this chair and going to bed. That'll be really my exit.
Well, Dr. Rydzak, I really appreciate your time today and for sharing your research with us. Absolutely, and I hope to keep in touch.
Yeah, man, absolutely. I hope to keep in touch as well.